Hello everyone, my name is Dewi Syahidah and I am a research staff at Pathology Laboratory, Institute for Marine Research, Aquaculture and Fisheries Extension, Indonesia. Today I would like to share with you about our study on susceptibility of raw blue interferon stimulated gene cell cultures to Chequa Iflavirus and Athtab bunyavirus from stressed Redclaw crayfish, Cherax quadricarinatus from the northern Queensland. This is the outline of what I'm going to talk today. First, I would like to give you a brief introduction, and then following that, I will give you some research questions from our study, and I would like also to describe about materials and methods that used in the study. And then I would like to continue with our results and discussions, and following that I will give you some conclusion of our study, and also I would like to give you some future direction of our study. This is the map of Australia. As you can see here, the yellow color indicates the central production area of crayfish aquaculture in the country. The crayfish aquaculture growing rapidly due to the species benefits. As the farming industry is growing, issues of crayfish diseases becomes more important. The discovery of Chequa Iflavirus and Athtab virus from Redclaw crayfish provide important insights into the potential diversity, taxonomy, and evolution within the family Iflaviridae and within the family bunyavirus. Relationships between viruses in insects and crustaceans have been reported. However, due to a lack of Chequa Iflavirus and Athtab bunyavirus-free crayfish, and with no cell cultures, therefore an alternative cell culture to propagate the viruses is needed. Now, these are some examples of previous reports from different study groups. These two papers here discuss about the two viruses isolated from stressed Redclaw crayfish in Australia. The viruses are Chequa Iflavirus and Athtab bunyavirus. Whereas this paper here discuss about a mammalian cell lines could be infected by a shrimp virus called Taura syndrome virus. This figure here is an example of Redclaw crayfish or Cherax quadricarinatus. Now, mouse macrophage cells, or what it is called as raw cells, is the most common cells used to study interaction between viruses and their hosts. Raw blue interferon stimulated gene cells originally were designed for the detection of bioactive murine type 1 interferon. Previous studies of using insect and mammalian cell cultures to understand the infection and replication of some crustacean viruses is still open to question. Now, from the background studies, and we came out with some research questions. Understanding mechanisms involved between viruses and the host had been hampered with some difficulties. Detection in low-level infection was considered less sensitive, therefore we need PCR. The major impediment to study the virus replication was considered lack of suitable cell lines. Therefore, we conduct this research study. Now, these are some materials and methods that we use in this study. We grew the cells in the MEM medium and we add serum about 10%. we used 6 tissue culture flasks and we divided them into 2 groups, infected and uninfected groups. We collect hemolymph of infected Redclaw crayfish with both Chequa Iflavirus and Athtab virus. We inoculated them in the 3 flasks, whereas other 3 flasks we use them as negative control flasks. We collect raw cell samples on different days post-infection. Then the cell samples were subjected to H&E stain and PCR. These are some primer sets that we use in our study based on the previous references. And then we conduct RT-PCR with 3-step cycling. Following that, we run gel electrophoresis. Now, I would like to move on to our results and discussion. This figure here illustrates the H&E stain of infected and uninfected raw cell culture. Based on this figure, we were very difficult to identify or determine the differences between uninfected and infected cells upon virus inoculation. Now, this figure here illustrates the RT-PCR of Chequa Iflavirus in raw cell cultures. If we have a look on this box here, we can see that the virus can maintain their survival until 1 week post-infection. On the other hand, this figure here informs us the RT-PCR of Athtab bunyavirus in raw cell cultures. If we have a look on this box here, we can see that the virus can maintain their survival until day 4 and then start to vanish on 1 week post-infection. This figure here illustrates the quantitative RT-PCR assay of Chequa Iflavirus. This is the amplification plot. This is the MELD data, and this is the standard curve from 10-fold serial dilution of linearized plasmid DNA containing Chequa Iflavirus insert. Now, from our findings, then we can conclude that the H&E stain not sensitive enough to detect viruses in our raw cell cultures. Athtab virus infect raw cell cultures, stays in the cells until day 4. Possible the stronger amplicons are due to the virus's immediate early gene RNA-dependent RNA polymerase on which the RT-PCR is based turn on the messenger RNA plus viral genome. Chequa Iflavirus infect our raw cell cultures and can maintain their survival in the cell cultures until 1 week post-infection. Now, from our study, we can then propose some future directions for the upcoming research related to what we have conducted in our study. First, we can evaluate the growth of both viruses in raw cell cultures using a qPCR. And then we can also design new PCRs to look at later gene and see if PCR amplicons increase. Or we can evaluate raw cell cultures for studying nuclear location signals of crustacean viruses. We can also assess a mosquito cell lines, in this case C636, for in vitro propagation of Chequa Iflavirus. Now, these are some references that we use during our study. Thank you very much for your attention. Any questions so far?